Hello and welcome to G Fun Facts Online, the podcast that unpacks the incredible science behind our world and ourselves. Today, we're going on a journey deep inside our own bodies, into the very code of life, our DNA. Now, you might picture DNA as a static, unchanging blueprint, a fixed instruction manual for building you. But what if I told you that parts of this manual are alive, constantly cutting, copying, and pasting themselves into new locations? What if I told you these nomadic bits of code, once dismissed as useless genetic junk, are actually powerful architects of evolution, sinister drivers of disease, and now, one of our most promising tools for curing those same diseases? Get ready, because today we're talking about the incredible story of transposons, better known as jumping genes. Our story begins not in a modern high-tech lab, but in a cornfield in the 1940s, with a brilliant geneticist named Barbara McClintock. While studying maize, she noticed something odd. The corn kernels had strange, variegated color patterns, splotches and streaks that defied the rules of genetics as they were known. Genes were supposed to be fixed points on a chromosome, but McClintock hypothesized something radical, that certain genetic elements were physically jumping around the genome. When one of these elements jumped into a gene responsible for color, it switched the gene off, creating a colorless patch. When it jumped out again, the color came back, creating a colored spot. This idea of mobile genes was so revolutionary that the scientific community just wasn't ready for it. Her work was largely ignored for decades. It wasn't until the 1970s, when similar elements were found in bacteria, that the world caught up to her genius. In 1983, Barbara McClintock was finally awarded the Nobel Prize, a testament to a discovery that completely rewrote our understanding of the genome. So how exactly do these genes jump? Well, they have two main strategies. The first is called copy and paste. This is the preferred method of the most common type of transposon in our bodies, the retrotransposon. It works by making an RNA copy of itself, then using a special enzyme to convert that RNA copy back into DNA, which is then pasted into a new spot in the genome. The original copy stays put, so the number of these transposons just keeps growing. In fact, over 10% of your entire genome is made up of just one type of these copy and paste elements, known as ALU elements. They are essentially genetic parasites, relying on other elements to provide the machinery they need to move. The second method is more straightforward: cut and paste. An enzyme, often made by the transposon itself, snips the gene out of its location and pastes it somewhere else. The original is gone, simply relocated. For years, scientists saw all this jumping activity and labeled these transposons junk DNA, thinking they were just useless, selfish bits of code along for the ride. But we now know they are one of the most powerful engines of evolution. By jumping into new locations, they create genetic mutations. Most of these are harmless or detrimental, but every now and then, one is beneficial, creating a new trait that natural selection can favor. They can also carry control switches with them, landing next to a gene and changing how or when it's turned on, creating complex new genetic networks. Some researchers even believe that a massive burst of activity from these jumping genes, specifically the ALU elements, in our ancestors millions of years ago, coincided with the rapid expansion of the human brain. This flood of new genetic material may have provided the raw material for the evolution of our unique cognitive abilities. But there is a dark side to these genetic nomads. While they shape species over millennia, their jumping can be catastrophic for an individual. When a transposon lands in the middle of a critical gene, it can disrupt its function and cause disease. The link to cancer is becoming alarmingly clear. In many tumors, the cellular mechanisms that normally keep transposons locked down fail, allowing them to jump around again. They can jump into tumor suppressor genes, disabling the cell's natural brakes on growth. They can land next to a cancer-causing gene, acting as a rogue on switch. This chaos contributes to the massive genomic instability that is a hallmark of cancer. The brain is another hotspot. In devastating neurodegenerative diseases like ALS and FTD, some patients experience a transposon storm, where these elements become hyperactive, contributing to the death of neurons. Links are also being found to Alzheimer's, schizophrenia, and autism, and sometimes, a single jump is all it takes. A transposon insertion into the right gene can be the direct cause of diseases like hemophilia and Duchenne muscular dystrophy. So they're architects of evolution, but also agents of disease. It's a classic Jekyll and Hyde story. But here's where the story takes another incredible turn. Scientists are now taming these jumpers, turning them from a source of disease into a revolutionary medical tool. By engineering transposons, we can now use them to perform gene therapy. Systems with cool names like Sleeping Beauty and Piggyback allow us to cut out a faulty gene in a patient's cells and paste in a healthy therapeutic one. This is already being used in clinical trials for CAR T-cell therapy, a groundbreaking way to engineer a patient's own immune cells to fight cancer. The future of this technology is even more mind-blowing. Scientists are combining the power of jumping genes with the precision of CRISPR gene editing. Imagine a technology that uses CRISPR to guide a transposon, carrying a large therapeutic gene to a very specific, safe location in the human genome for a permanent fix. This isn't science fiction. Systems named Cast and Create are being developed right now to do exactly that. From the colorful, speckled kernels of corn studied by Barbara McClintock to the forefront of 21st century medicine, the story of the jumping gene is a powerful reminder of how much we still have to learn. What was once dismissed as junk is now understood as a dynamic force of nature, a creator, a destroyer, and now a potential savior. It proves our genome is not a dusty old book, but a vibrant, living library with secrets still waiting to be read. Thanks for tuning in to G Fun Facts Online. Join us next time as we explore another marvel of science.